Hey everyone, did you know that conventional wastewater treatments fail to eliminate emerging contaminants, leaving trace concentrations of hazardous pesticides like Diuron to persist and pollute aquatic ecosystems? The researchers, Helvig and colleagues, hypothesized that genetically engineering bacteria, Bacillus megatherium, and Microalgae, Chlamydomonas rehanetide, to express a mutated P450 enzyme, P450 BM3 MT35, could metabolize the targeted pesticide Diuron in wastewater. While one-type microorganism can degrade Diuron naturally, this process often takes weeks, far exceeding the retention time in municipal wastewater facilities. This study aims to fill the gap by engineering low-cost microorganisms that can detoxify emerging water contaminants. The study utilized a multi-organism approach. The mutated enzyme P450 BM3 amplified was first expressed in E. coli for in vitro purification in assays. Then, the gene was transformed into B. megatherium via protoplast transformation and the chloroplast of C. rehanetide algae for in vivo metabolism studies in both synthetic and municipal wastewater environments. Subchronic and histec purification, HPLC, and mass spectrometry to track Diuron degradation and metabolite formation, and the use of the P450 inhibitor larozole to confirm the specific enzymatic activity driving degradation. The bacteria experience reduced Diuron metabolisms in static municipal wastewater due to nutrient limitation and suboptimal growth conditions. The authors also note biosafety challenges regarding the uncontrolled proliferation of genetically modified organisms in open environments. P450 BM3 successfully degraded Diuron into two primary metabolites, DCPMU and DCPU, without forming toxic degradation product 3,4-DCA. This figure shows the HPLC chromatogram represents peak retention times corresponding to Diuron, and the three Diuron metabolites, DM1, DM2, and DM3, were observed when incubated with the purified P450 BM3 with and without NADPH. This chromatogram provides direct evidence that P450 BM3 successfully degrades Diuron into less harmful metabolites. Transgenic D. megatherium degraded 65% of Diuron after 5 days in ideal broth media and achieved 45% and 50% degradation in synthetic and municipal wastewater, respectively, while type bacteria showed no metabolisms. In Figure A, the open squares represent the amount of Diuron remaining, while the triangles represent the formation of Diuron metabolites. Figure B compares the mutant enzyme with the wild type P450 BM3. Figure C examines how environmental conditions affect degradation efficiency. After 7 days of treatment, the two engineered strains CRM1 and CRM2 degraded approximately 50.8% and 53.3% of Diuron respectively. In contrast, the wild-type algae degraded only 6.9% of the herbicides, indicating very limited natural biodegradation capacity. The engineered algae were able to tolerate higher concentrations of Diuron. The transgenic strain remained active even at concentration that inhibited the growth of the wild-type algae. Previous approaches, microbial consortia and natural biodegradation systems, require several weeks to months to achieve substantial Diuron degradation. Although methods like immobilized enzyme and photodegradation can achieve high degradation efficiencies, they are expensive and difficult to scale up. In contrast, the genetically engineered bacteria and microalgae developed in this study achieved approximately 65% degradation in bacteria within 5 days and over 50% degradation in algae within 7 days. The researchers then added larozole, which is a P450 enzyme inhibitor, to determine whether P450 BM3 was truly responsible for the enhanced degradation. Without larozole, the transgenic algae showed improved Diuron degradation and were able to tolerate higher concentration of the herbicide. However, once larozole was added, the enhanced degradation disappeared, resistance was lost, and the transgenic algae behaved similarly to the wild-type strain. The enzyme P450 BM3 successfully enhanced both Diuron metabolism and organismal resistance. First, the engineered microorganism showed efficient degradation of Diuron. Second, not highly toxic 3,4-DCA was detected during the degradation process, suggesting that this approach may be safe. Finally, the engineered organism showed improved tolerance to Diuron exposure, meaning they would survive and function in the higher herbicide concentration. One of the most interesting findings of this study is the use of microalgae as bioemulsion platform. Microalgae naturally perform photosynthesis, which continuously produce NADPH. And NADPH is the reducing power required for P450 enzyme activity. Therefore, the algae can provide their own energy source to drive pollutant degradation without requiring external cofactor supplementation. Engineered organisms could be integrated into real-world wastewater treatment infrastructures such as high-rate algae ponds, biofilters, or controlled bioreactors to act as a tertiary treatment step for pharmaceutical and pesticide removal. The author suggests conducting pilot-scale experiments in controlled wastewater treatment plants to test scalability and enzyme stability alongside exploring regulatory frameworks and biosafety containment strategies like algae immobilization. The study shows that Diuron degradation from in vitro enzyme assays all the way to in vivo applications, the researchers successfully engineered both bacteria and microalgae. The study use of the P450 inhibitor, lerozole, to confirm the role of mutant enzyme. The researchers tested the engineered bacteria in actual municipal wastewater. Despite these trends, several limitations remain. The expression of P450 BM3 amplified in bacteria appeared to be transient. A third minor metabolite, known as DM3, was detected but remained unidentified. The study also did not investigate the long-term stability, and no pilot-scale experiments were conducted. The author suggested that the metabolite DCPU and DCPMU appear less toxic to the engineered algae. There is also the possibility of long-term accumulation of these metabolites in the environment. Therefore, additional ecotoxicological studies are needed before this engineered microorganism can be safely implemented in real wastewater treatment systems. In conclusion, this study demonstrates that genetically modified bacteria and microalgae equipped with the P450 BM3 amplified enzyme provide a cost-effective, rapid, and targeted alternative for removing persistent contaminants that conventional wastewater treatment systems cannot efficiently eliminate. This study proved the viability of using advanced transgenic chassis organisms, especially photosynthetic algae, for sustainable, large-scale environmental bioemulsion of industrial and agricultural pollutants. As we develop highly efficient engineered microbes to detoxify our wastewater, how do we best balance their immense bioemulsion potential with the ecological and biosafety risks of releasing modified organisms into the environment?